take a look at the physiology of the kidney, which is primarily the formation of urine. So about 180 liters of fluid is processed daily. However, only about 1.5 liters of urine is formed each day. So that's going to give you an idea of the basic underlying premise of how we make urine. We're going to put a lot of fluid in, and we're going to take most of it back out. So a lot of filtrate, little urine, only about 1% urine. And the kidneys are filtering the body's entire plasma volume about 60 times each day. They consume 20 to 25%, almost a quarter of the oxygen we use in our bodies when we are at rest. They are producing filtrate by glomerular filtration. That is basically blood plasma without the proteins. The urine will be produced from the filtrate. So urine makes up about 1% of the original filtrate, and we want it to contain water and the metabolic waste and the other unneeded substances, not everything in the plasma, the way that filtrate does. There are going to be 3 processes involved in the forming of urine and adjusting that filtrate so that we can maintain blood composition. Those 3 processes are glomerular filtration, which produces cell and protein-free filtrate, tubular reabsorption, which is going to selectively return about 99% of the substances from the filtrate to the blood in the renal tubules and collecting ducts, and then tubular secretion. That is to get rid of substances that are still in the blood that need to be removed, that didn't get into the filtrate to begin with. So we've got glomerular filtration, tubular reabsorption, and then tubular secretion. We're going to look at all 3 of those as independent sections. Starting with glomerular filtration. Glomerular filtration is a passive process. It does not require metabolic energy. So no ATP is involved. There would be hydrostatic pressures that are going to force the fluids and solutes through that filtration membrane we looked at in the first part, and it will end up in that glomerular capsule. We're not going to have any reabsorption here. It's pushing fluid out, and that fluid collects in the capsule and makes its way through the tubules we described before. That porous membrane between the blood and the interior of the glomerular capsule allows water and solutes smaller than plasma proteins to pass through that. Normally, we do not let cells through there. It's too small for cells. It contains 3 layers: that fenestrated endothelium of the glomerular capillaries, a basement membrane, which is the fused basal lamina of the 2 other layers, and the foot processes of the podocytes with filtration slits. And those slit diaphragms are going to repel large molecules, macromolecules, such as proteins. So here we have the arterioles coming in. They're surrounded by these podocytes, and fluid without proteins in it can make its way into this capsule and then into the proximal convoluted tubule. So this is where filtrate is formed. And if we look closer at those, you see the way the podocytes wrapped around them and the little foot-like extensions making filtration slits. And this is the same thing with an electron microscope. So plasma in the blood makes its way into through the filtration slits, and over here it becomes filtrate. Macromolecules get stuck in the filtration membrane, and they would be engulfed by glomerular mesangial cells. This allows water molecules and other things smaller than 3 nanometers to pass. Those kinds of things would be things like water, glucose, amino acids, nitrogen waste. Plasma protein should remain in the blood. That helps to maintain colloid osmotic pressure so we don't push all the fluids out of the circulation. at one trip through the glomerulus. And proteins if we do find proteins in the filtrate, that would mean there is a problem with the filtration membrane. So the pressures that will affect filtration include the outward pressure. That's the force that promotes the formation of filtrate. Hydrostatic pressure in the glomerular capillaries is essentially glomerular blood pressure. It's the chief force that's pushing water and the solutes in it out of the blood. It is quite high, about 55 millimeters of mercury. Most capillary beds have about half that, 26 millimeters of mercury. And the reason for that is that the efferent arteriole is a high resistance vessel with a diameter smaller than the afferent arteriole. The inward pressures, so that we don't push everything into the capsule, is the hydrostatic pressure in the capsular space, that filtrate pressure in the capsule. That adds up to about 15 millimeters of mercury. And the colloid osmotic pressure, that's the pull of proteins, the difference in osmolarity between the two places, and that is about 30 millimeters of mercury. So our net filtration pressure would be the sum of all those forces, 55 forcing out, 45 opposing, so a net outward force of 10 millimeters of mercury, and that push of 10 millimeters of mercury is what we use to push the filtrate into the capsule, and that is the major controlling factor that determines something called glomerular filtration rate. That glomerular filtration rate is the volume of filtrate formed per minute by both kidneys, and normally that should be about 120 to 125 milliliters per minute. It is directly proportional to the net filtration pressure. That is the primary pressure in the glomerular hydrostatic pressure we just went over. It is also directly proportional to the surface area available for filtration, so that would be controlled by glomerular mesangial cells that can contract, and the permeability of the membrane, known as filtration membrane permeability. And these capillaries should be much more permeable than other capillaries. A constant GFR is important because it allows the kidneys to make filtrate and maintain extracellular homeostasis. There are intrinsic controls over it, and those intrinsic or renal autoregulations maintain the GFR in the kidney. And GFR is going to affect systemic blood pressure. So an increased GFR is going to cause an increased urine output. That would lower blood pressure by removing fluids, and vice versa. The goal of extrinsic controls, controls from outside the kidney, is to maintain systemic blood pressure, and that would be the nervous system and the endocrine mechanisms that are working on the kidneys. So intrinsic controls, regulation by the kidneys themselves, renal autoregulation, works to maintain a nearly constant glomerular filtration rate when the mean arterial pressure is in the range of 80 to 100 millimeters of mercury. If we go out of that range, below or above, autoregulation won't be able to keep a constant GFR. The two types of renal autoregulation include myogenic mechanisms. Myo should tell you muscle right away, and tubuloglomerular feedback mechanisms. The tubules talking to the glomerulus. So the myogenic mechanism there, smooth muscle contracts when stretched, increases blood pressure. That would cause an increase in blood pressure, leading to a constriction of the arterioles that would restrict blood flow into the glomerulus and protect the glomerular from high blood pressure damage. A decreased blood pressure would cause dilation of the afferent arterioles, and those are going to work together to maintain a normal glomerular filtration rate even though blood pressures are changing. The tubuloglomerular feedback mechanism, the flow-dependent mechanism that's directed by macula densa cells. They respond to filtrate salt concentrations. If the GFR increases, that filtrate flow rate would increase. That would lead to a decreased reabsorption time, which unfortunately we'll talk about later on, but that an end result of that is it causes high salt levels in the filtrate. So a feedback mechanism takes over that causes the constriction of the afferent arteriole. That would lower the net filtration pressure. That's the glomerular filtration rate. That would allow more time for salt reabsorption, and we would have the opposite mechanism if we have a decreased glomerular filtration rate. Extrinsic controls from outside the kidney, we have neural and hormonal mechanisms. Their purpose is to regulate GFR in an effort to maintain systemic blood pressure. Extrinsic controls can override renal intrinsic controls if blood volume needs to be increased. So the sympathetic nervous system under normal conditions, at rest, the renal blood vessels are dilated and renal autoregulation prevails. Under abnormal conditions, such as an extremely low extracellular fluid volume, which would mean low blood pressure, norepinephrine would be released by the sympathetic nervous system and epinephrine by the adrenal medulla. That would cause a systemic vasoconstriction. That would increase blood pressure. It would also cause constriction of afferent arterioles, which would decrease glomerular filtration rate, causing the net effect of having blood volume and pressure increase and the sympathetic nervous system controls. The renin-angiotensin-aldosterone mechanism, this is kind of the hormonal, is the main mechanism for increasing blood pressure. And there are three pathways to renin release by the granular cells. We can get direct stimulation of granular cells by the sympathetic nervous system. Get stimulation by activated macula densa cells when the filtrate salt concentration is low, or the reduced stretch of granular cells. And that is mapped out here in this image, and I again encourage you to go to 2514 and follow this I forget what I want to call these, flow charts, I guess. Other factors that affect the glomerular filtration rate: the renal cells release a variety of chemicals, and some of those act as paracrines that affect the renal arterioles. Adenosine will do that. Prostaglandin E2 will do that. And some cells make their own locally acting angiotensin II, and that reinforces the effect of hormonal angiotensin II. So we have the intrinsic controls and the extrinsic controls compared. Anuria is the abnormal low urinary output, and that would be clinically defined as less than 50 milliliters per day. That could indicate that the glomerular blood pressure is too low to cause filtration. Renal failure and anuria can also result from situations in which the nephrons stop functioning. So acute nephritis, or inflammation of nephrons. Transfusion reactions can do that. They clog up the filtration membranes, and crushing injuries to the kidneys can do that. All right, we'll pick it up